So that as in like a title that here show like let me see here. Okay, later pointer. Okay, so there are here you see that like a Hotinia cordata, like a Korean name Osongcho. Many of you know about that are like some like a key like a material that are utilized for the like Oriental medicine for the patient, and we generate that is a name core extract from this Osongcho that TDN named that TDNA0101. Then I utilizing this like the compound to testing the like effect on Alzheimer disease. So utilize with coupled with my technology. Okay, so
Firstly, I disclose that I don't have any financial commercial interest on the with the pharmacovarium, which hold patent for the PDN0101. So first of all, let me
start with the this what is core extract. This the compound 0101 that it contains 5 major components of it. But let me just here mention 2 of it. One is like here the oh sorry. Okay, good. Okay, lamnogalactunan. 01, this like 0101 is like active polysaccharide that contains these 2 major, around 70%, over 70% of this one. One is lambda glucuronan that, and the other one is constituent. One is first one that is like both of these like a lot of research out there available that study in the first one in case of lambda glucuronan that having some tight junction that benefit and like a mucosal like immunity benefit and so on. So all these kind of things that are like we imagine that are like a gut-brain axis or impact on the gut immunity. And second one is positrin. That positrin is, as you see here, the second, the bottom here, the positrin is small compound that it has like an antioxidant or like an anti-inflammatory effect, as well as the anti-diabetic some effect, like a benefit on the like when they like using this kind of component of it. So this kind of like 0101 has some of like we expect some of benefit of it. So then. Like, let me just move to what is Alzheimer. So many of you know what is Alzheimer disease. So that I just mentioned here about what type of study I do, what I know. In my point of view, what is Alzheimer disease? Alzheimer disease is progressive neurodegeneration, which that classify Barak stage internationally that name that are like a common that stage of like a disease. From the Barak stage 1 to 6, so initial like abnormality or cellular dysfunction in the entorhinal cortex and or prefrontal cortex. That's the early stage they named, and the second like in the they transit to the middle stage, which the area affecting the hippocampus, and then the late stage that are like a neocortex, all the brain area affected by something the symptomatic effect that are like a dysfunction of cellular, like a cellular and physiological dysfunctions. And as you know, this Alzheimer disease is a memory loss, and with a massive brain, like a loss of brain volume, with a like a cell death. And then this call is we see there are like a progressive neurodegenerations. So that here, definitely, like most of you know that are like a death, kind of like a cellular death or cellular toxicity, starting from the protein aggregation that such as amyloid beta or phospho like tau proteins. And these kind of things that kind of like affecting some of like one of the recent publication in 2024 that CGS STING pathway that affecting the activation of microglia. And then this is one of like potential mechanisms that are associated with the Alzheimer. Then in this case, like A1 astrocyte conversion of A1 astrocyte that are like that's 2000. I remember that 2017 that published from our group that like conversion of A1 astrocyte that the affecting that are drive that are like a neuronal cellular death, and this kind of like induce that are like kind of neurons that some of functional neurons that drive. And then some of mitochondrial dysfunctions or kind of like a metabolic disorder or vascular damage, all those are definitely affecting symptom effect and Alzheimer's disease. And definitely some of like a really low percent of it, like a 2 to 3%, but the genetic mutation drive that strongly drive that are like some such as like ApoE proteins that drive that are like Alzheimer's disease. So we tried 0101 on the all laboratory, many of the laboratory try kind of evaluating their effect using utilizing kind of like some staining and a tissue like histochemistry staining and a cellular staining, and definitely we tried that like a very basic assessment to the evaluating this kind of like a protein aggregation. As you see here, that are like in the right bottom, that with the staining picture with that, like 0101, like strongly like inhibit the aggregation of that are like this phospho-tau and neurofibril tangles. And this was generated by one of like our undergrad student that the definitely is like measuring. Those are like phosphoproteins associated with the immune activation. It clearly show that are like, oh, that's inhibited or like a pretty good evidence and are like easily repeat that kind of like some who are like on like some that are like who is like a professional, even not a professional, easily replicate the data. And the other one that we also evaluate kind of like whether this 0101 that help like inhibiting the conversion of A1 astrocyte when we treat with the treatment of 0101, we see that are like a very significant and clear that this 0101 like inhibit this conversion of A1 astrocyte. Or this effect, we definitely extend our research to the embryonic stem cell derived astrocyte and microglia and neurons. All this show that are very similar. There are level of that inhibiting. Those are like inhibiting the toxicity that of like kind of like some A beta and other like drivers. Okay, then we name that. Okay, 0101 solve everything. Then what like we call that are like okay multimodal pharmaceutical effect. In the Korean, like we call 다중기작, right? So there are like 0101 exhibit significant mechanistic effect in multiple AD related pathway. But we have many of you meet kind of like a challenge, this type of challenge. We know where we see that or like we haven't seen this kind of like a very significant effect on the like amenorrhea this AD pathology, but they are like to get the some approved as a drug on this kind of 0101 natural compound is super challenge. So there are one of the things that I believe if we identify the accurate mode of action MOA that clearly help to approach like a step forward to get the approved in as a like drugs. So then I utilize one of my technology that like I'd like to introduce. Many of you already know it's one of very common technology is a single cell. So yes, it's a simple. There are like if previously like the people handling here, there are like instead of processing the bulk average that information, we are trying to use dissect into single cell level, and then we get obtained that like collecting that are like information from individual cell or cell types, and then processing with some that advanced like computational algorithms. Here there are most recent, early this year, we like now under review that we developed in my laboratory that developed a new technology, single cell technology, can like couple with like a Cas9 that Cas9 and like depleting there are noise of like sequencing and so on. That are like we like develop a really high level of like high resolution of sequencing technology. So this point, let me just switching gear. Because let me just stop there talking about 0101. Just from now on, I'm going to introduce about my lab. And my lab study about big data. The other terms that we study is progressive. Okay, let me give you one question. About I'd like to share about big data. What's the big data for me? Everyone knows it's big data. You have a big data, you have your own big data, you have your own big data. So what is the big data for me? Okay, which is the American favorite pie? Strawberry, raspberry, blueberry, apple, mango, vanilla, chocolate. So I'm counting 1, 2, 3, then you're going to answer, okay? So question again, question is, what is Americans' most favorite pie? Raspberry, blueberry, something that apple, mango. I'm counting. You're all ready, right? 1, 2, 3. Yeah, that's interesting, yes. Yes, apple. Yeah. Answer is apple. How do you know that? Actually, you're right. Like an apple, you hear that many of answer say apple. How do you know that? Yes, data. Data from supermarket sales, Walmart and the Target. 18-inch of pie. That data say apple. Yes, no other competitors. Apple. Let's imagine this. When your parents done their day jobs and back to home, he wanna stop by supermarket and pick up one pie or pizza that everyone's favorite. However, that's another say. That is everyone's second favorite because your father or mother think about pick everyone's family members their favorite. But once starting there are supermarket sales there are smaller piece of pie. Data say number one is not apple. Because from now on, once there, you see there are from bakery, you see there are a small piece of pie, some many different choice you have. That case, you're going to choose one of your favorite, your first favorite, which means data that are big data, large big data that are just simply you're collecting more data. The others say you are same size of a data, but you can dissect into this smaller and smaller. Then you will see something better or something new you never seen before. So this is kind of the idea that are rather than processing that are all that are combined some average and handling processing that are everyone's second favorite a symptomatic target whatever diagnostic target druggable target second favorite. We are thinking about processing an individual sorted everyone's first favorite. In other say in the medicine precision medicine we are thinking about. The other terms in my lab study, progressive. We are, in a first slide I mentioned, we are working on the progressive neurodegenerations. And then we have to thinking about whether my research reflecting this progressive. Here, there are I using editing Photoshop that are this is here there are the highway is 15. Is heading to the way to the Las Vegas. Everyone getting age and a neurodegeneration. There is no exit. Everyone have to stay on this heading to Las Vegas for your life gamble. We don't know which type of disease we are going to have. Here there are most of number of patients Alzheimer's disease, most a large number of car in the street, but in the highway, but is no exit. But here we see this is a snapshot. But really, actually, this car is running 60 miles per hour, 100 kilometers per hour. But we are a research. It's like this, kind of like a snapshot. In a young blood, a young researcher nowadays, they like YouTube, not pictures. But whether you're like a research reflecting kind of like, in other say, fluidics rather than static. So there are like in a single cell, coupled with the single cell technology, two ideas we're thinking about. One is introduced in a Nature Biotechnology 2017. The other one is 2018 in a Nature. One named that trajectory. That as you see here, that picture you see is someone dived from the top of the mountain to the water. You can like say that like this kind of early time point that are like early stage, buttock stage 1 or 2 from the jumping point, and then this is stage that 5 or 6 who like very severely that progress than a disease. But like this case, how about processing the single cell? Like it's a missile that once missile have arrived, that are like immediately army is like calculating where the origin from. That we idea that bring that this kind of terms that in the algorithm on this like data analysis, so that we are tracing them, but we don't know how long does it take under pseudo time. So we name that pseudo time. We don't know exactly how long it's take a time, but because single brain, okay. Someone has like a Parkinson's disease or Alzheimer's disease, they have all the track information from the like some of progress of disease. Some cells are like severely affected, but some cells now under like starting there are like a kind of like some affecting there are like some of symptomatic effect. The other one is RNA velocity. It's utilizing some of like a part of central dogma that they measure. There are like unspliced splicing RNA to the splicing, spliced RNA, virtual RNA, and then degradation. Because RNA is sick from the nucleus, that we can measure the ratio of it. So it's kind of like you see that a picture. Once you open a little bit longer, open your shot, that you're taking kind of like a blur shot. Oh, so this is a bad picture. But the others say, you see the picture, where they going, we see, tracking them. If you like expose longer, you see that which pathway or which domain, taking all this mainly pathway that are like here. So here the example. This is an example that I just hear that from the other seminar that who presenting in the Parkinson's disease. That this is I utilizing as a proof of concept that utilizing inject of alpha-synuclein into the mouse brain that induce that Parkinson's disease. I'm not saying that like Parkinson's disease like model. Then I hear the control mouse brain. You see the 4 branches with the transition here. You see the length and branch that indicating that here the total like a transcriptional changes, and a branch means that kind of like transition or functional changes. So when they inject alpha-synuclein that around 4 months later, you see that like a kind of like their transition and their branches is totally changed. Then we assume kind of there's a point of change, the branch and the range transitions that associate kind of like a diagnostic marker. We can like have an opportunity to finding the diagnostic marker or druggable target if we have a chance. And the second one is RNA velocity. It's pretty much as you see in the picture, like our previous picture, there is just small area of it. As you see the arrow of I extract from the data only the astrocyte. And then when I processing it, it's type of machine learning algorithm that based algorithm. You see that a transition of A1 astrocyte. We clearly see their conversion of it with under alpha-synuclein exposure. Yeah. So I'd like to back to the 0101. So we cannot cover their entire brain after treatment of like oral garbaging that 0101 in 12 weeks. So that we need to decide which brain area we are going to cover. So we perform 4 different behavior tests. with a memory association. You know, there are like many of you know, there are like each of like a behavior test. I'm not go deep into this each of like a behavior test, but indicating this each of like the behavior test, they have a different like brain area that associate, so that we after performing and then like it's striking 0101 help like enhance their memory, all the memory tests, and then we giving the weight of each of the tests, and then we narrow down and we make the conclusion on the 0101 drive in a prefrontal cortex and hippocampus, which the area maybe 0101 this like a core extract drive maybe mostly impact, like a mainly impact on the early stage or mid stage of Alzheimer's disease. So we process 430,000 of single cell from the, actually we covered the more than 1 million cells, the single cell. And then like we process like only utilizing 430,000 high quality cell. So we need a supercomputer to processing in the same time. So we using the like Johns Hopkins, the high performance computing system. So here then, like from the data, single cell data, we just focusing on today. I'm just focusing on the microglia in a prefrontal cortex. I believe anyone who study in the like a microglia, you may remember the one of cell paper published Weizmann Institute of Science from Israel in the Cell 2017. What they like their like a highlight of their finding is a microglia in the normal condition, the homeostatic cell condition, like a subtype, stay, and once starting that triggering that some like toxic factors, then initiating the disease associate microglia subtype 1, and then this severity drive like a drive and to the conversion of this DEM1 to DEM2. So that this accumulation of this DEM2 giving that like it drives cellular death, neuronal death. So this is like interesting site, like last 7 years cited more than 45 around, like I think it's increased more than, now it's over 4,500 citation. That's amazing. That's amazing finding. So that we reprocess after 0101. Okay, let's see, in the control, and then Alzheimer model. Here is no 0101. In the control model, you see microglia here. Majority, you see they are like homeostatic. Yes, that's what we expected. And then in the Alzheimer model, you see in the DEM2. Okay, that accumulation of DEM2 cell types, inducing the cell death. That's why there are like a behavior test, the memory test showing there are like most of mice and they're like abnormal behavior in the behavior test. And then just here you see that compare the Alzheimer model with the 5xFAD model. Is a name like a most common using for the like a study in the Alzheimer, so that we treat like a 12 weeks of 0101. And clearly what you see, that's no DEM2 that abolished this 0101, that DEM2. And the other thing, we see there are something cell types here that we name it's bridge cell. We just first name the bridge cell. We see that something in the transition state, cell accumulating in the transition state. And the other one is, I think I'm missing the like a message from here. Weizmann Institute said homeostatic and convert microglia to DEM2 and DEM1 and DEM2. But here you data say, you see that in the like a trajectory model showing homeostatic goes DEM1 that are independent pathway, not in the like in the middle of pathway in the DEM2. And a DEM2 showing that are their own unique pathway. So which means we are like, we see that are like a little different type of microglia activation pathways. And with the treatment of 0101, we see there are like something that forming the bridge cells. Definitely we have a condition, just control. This is a really important experiment. Like people ignore something many times that are like not much focusing on the your kind of natural compound on the control conditions. You see here control condition and a control condition with the treatment of 0101, we see they're forming that, clearly see that a bridge one cell, which not observed homeostatic. Also treatment in the Alzheimer model, we see that the same thing forming the bridge cells. And this model we apply to RNA velocity again. Then you see there the arrow, the direction of arrow is go to homeostatic, which means this bridge cell not convert into the DAM1. It's not like another DAM1 type, which means homeostatic can convert to the DAM2, but bridge cell firstly go back to homeocell. Then after homeocell, maybe can have a chance to turn into like a DAM2 cell type, subtype. And not data is not showing here, but we just compare that or characterize about this bridge cell is very similar, that cells generate after you exercise. When you after you exercise, forming the micro cells, we compare their gene expression pattern of it. It's very similar with the bridge cell, which we believe this bridge cell can block, maybe can block their conversion or progression of neurodegenerations. Definitely this kind of a hypothesis. Yeah, last couple slides left. Okay, thanks. That we review that are earlier published in a 2003 Cell paper. They're kind of consortium. They processing the 427 Alzheimer patients in the single cell, and we found from the normal condition, and many of a non-disease control contains having this kind of bridge cell types. So here the last slide, summary of my observation at this moment. I'm not covered that, only covered today microglia in an early stage in a prefrontal cortex. I can definitely share with you about there are all different cell type or in the information. But today here conclusion, there are homeostatic cell. It directly convert to DAM2, not DAM1. DAM1 has another independent pathway, but we are not focusing on about this conversion. So. Can hard to answer what's happened there, but treatment 0101 induced this kind of bridge immune cell, which prevent that a preventive or protective effect on the progression of disease. And this mode of action, clear and accurate mode of action that we believe help multimodal, which that I presented first, 5 slides that are effect on the kind of immune, inhibiting the immune activation or astrocyte conversion or inhibit the formation of neurofibril tangles. That's multimodality in Alzheimer model. That's upstream. I'm going to say it's master regulating cell in the potentially in the Alzheimer disease. Yeah, so that as I mentioned again, that I really want to see that whether acupuncture acupuncture therapy can induce kind of a continuous treatment, or this treatment can induce kind of bridge cell health that are improve their health on this kind of things, or some of who handling their natural product. That case, I'd like to collaborate and I want to see that whether this has a similar type of effect, or have a unique pathway on their treatments. Yeah, thank you for here that it's really glad that actually Hee-Jin Jo from Kyunghee University, and she joined a postdoc in 3 years in our lab. That she here the picture. And she mainly lead this project. And the 2 computational scientists, Chin Park and Dong Sang Kim from KAIST, actually they are computational scientists. They don't have any background on the biology. They are have like a computation scientists and many others that for collaborating. And especially, I really thank and glad to my parents in my first time presenting my front of my parents. I'm really glad, and I hope you also having in future have a chance to this kind of pleasure so. And again, that this is the first taking the GTX. I'm also taking my first time in the GTX with my mother. So all this makes this ISAMS that make me giving me all their personal experience. Personal experience you never forget. So I believe appreciate it organizing all the committee and the leadership. So thank you. Yeah.